So now we are going to talk about, spoken about the different architectures, transmitted light versus fluorescence, and the different types of microscopes. And now we're going to talk a little bit more into details about the different types of microscopes. So we're going to have wide field and single point confocals. So what is the difference between these two? So I think 100% of the students here are in one or the other situation, wide field or single point. So I remind you that on a big system like this, we have the microscope body, and then we have all the satellite equipment. And then what we're interested in is in identifying the equipment that is attached to the microscope body via hard connections, okay, to the microscope body or between themselves like this, via hard connections. So what can they be? They could be cameras, so it's one type of sensor. We could have scanners, so cameras or detectors, so it's the sensors, or we could have scanners, or we could have a few more things. So let's have a look at that. So again, there's only two types of microscopes, those without a camera, and so this would be single point confocals, and those with a camera, and there's all the others. And they have either, if they do not have a scanner, they're wide field, and if they have a scanner, they are multi-point confocals. So I do not think that anyone in this course has a multi-point confocal. It's unusual. Usually we always have a few. And all modern microscopes acquire digital images anyway. So the sensors are always acquiring digital images. So let's have a look at what a digital image is. So if we see this image of the edge of a cell, the image is a matrix of picture elements. This is what pixel means. It means picture elements, some sort of a contraction. And each pixel has an XY in coordinates and an intensity, okay? The intensity is not just any type of intensity. They're always a finite number of possible values, and these are always integers. So you cannot have an intensity of 2.3, okay? And then you cannot have, if you have an 8-bit image, and we will talk about the bit depths next week, if we have an 8-bit image, you can only have intensities that are between 0 and 255. So we have 256 integer values, okay? So this is what the intensity is. So when we zoom into this image, we could take this sample, acquire this sample in different ways. We can decide ourselves if we want to use small pixels or large pixels. And but in any case, we always have this little square in the image. And if you look a little bit closer, you will see that each of these squares actually corresponds to one X, one Y, and one uniform intensity. So throughout the square that represents the pixel in the image, we have only one intensity. And that means that we must have a device that records the XY position and a device that records the intensity. In any case, when we think about an image, we do have millions of pixels in an image, but we do not have only the X, Y, and intensity information in the image. We have also something called metadata, which is information about the way the image was acquired. So the metadata is very important. If you remember, when you applied to the course, I asked you to upload some images, and I insisted that they would be in the original format. And why? That is, if you do not use the original format of that the manufacturer recommends, you have a high risk of losing the metadata. So you keep the pixel information, so the X, Y, and intensity, but you lose the metadata. And the metadata is very important for you to come back to the image later and be able to say which objective you've used, which filter was set, etc. At which Z level were you, and everything. And when you go to your microscope, you open an image. On many microscopes, you're able to push some sort of reuse button that sets the microscope microscope, again, exactly the same way you had the microscope when you acquired that image, and this is because the microscope reads the metadata and applies all these settings to the microscope. So the metadata is very important, and it's important that it's preserved in the image. It's embedded in the file, so it's a single file. Okay, you have only one file most of the time. So we're going to acquire images with different sensors, with or without a camera, but anywhere we're always going to have X, Y, and intensity information. If we have a microscope without a camera, so it's only one type, is single point confocals, and these sensors are called detectors, okay? Or at least this is the way we're going to call them here. There are lots of different names. There's only one pixel, so each detector acquires only one pixel at a time, right? Just one pixel. And the detectors always record images in black and white. There's no such thing as a color detector. We're talking about each detector. We will talk about spectral detectors later, but even in spectral detectors, each detector only acquires black and white images. We can have up to 34 detectors per microscope, so that's quite a lot. But in any case, when we acquire all these with all the 34 detectors, is only 34 colors of a single pixel, so only one pixel at a time. And we will see all this in the lecture called Imaging Multiple Colors at Once, which comes next week. So the detectors only record the light intensity. And so we must have, so only the intensity. We need to have a scanner which records the XY coordinates. Okay, so when we use a single point confocal, the detectors only record the intensity of one pixel at a time, and then the scanner records the XY coordinates. The name of the detectors varies, and you may even have in each of your microscopes, you may have different types. So we can have conventional PMTs, GASP PMTs, HIDI, APD detectors, etc. So you may find these names in the software of your microscope. You will have to tell me which detector you have. So this is why I'm mentioning these names to you, so that you can recognize when you can recognize them when you look at your software. So what about the microscopes with cameras now? So this is all the other types of microscopes use cameras, wide field, multi-point confocal, light sheet, etc. Oops, what was that here? I went the wrong way. And the sensor is called a camera. And the camera acquires several millions of pixels at once, at the same time. Okay, so it's very different from a single point confocal, where the detectors only acquire one pixel at the same time. The cameras can be either black and white or color cameras. We will talk about all this a little bit later. And you can have up to 4 cameras per microscope, and that means 4 images of millions of pixels, okay? And then the cameras record both the light intensity and the XY coordinates. So it's quite different. That's one of the major differences between the single point confocals. The detectors record the intensity, the scanner records the XY coordinate. On the camera-based system, the camera does the whole job. Intensity and XY. And then some systems with a camera work with scanners and some don't. So we will see the difference. The name of the cameras they can be called CCDs, SCMOS, EMCCD, and they can be either front illuminated or back illuminated. So these are names that you may find when you search something you will have to do. You will have to find out which camera you're using, and you will have to Google some information about it, and you may find these names when you do that. So basically what we're looking at now is single point confocals and mostly wide field systems for in your case. So I just want to show you this, if it starts, yes, this little video for people who have never imaged with a point confocal system. When you image at high resolution with a point confocal system, this is what happens, because we image 1 pixel at a time. So you've just spent 23 seconds of your life in front of a point confocal. Now we go to camera-based system, blink, right? So there's an enormous difference between single-point confocals and camera-based systems. Is the camera-based systems, they acquire all the pixels at the same time. And that's not the case for single-point confocal. So here we go, we have a wide field system, and we see where I'm going to show you how a wide field system works. So we have a sample here that is labeled. Here we have the objective. This is an inverted microscope because the objective is pointing upwards towards the sample, so it's below the stage. And then the sample is in focus. So we have an objective focal plane that we're looking at. So currently there is no light. The sample is not lit at all. And here we have a mercury lamp or LED. And we could also have lasers in here. And then this contains lots of different wavelengths. And it's not, if it's a laser, then it's just one color. And then we have an excitation filter. And then when we have the excitation filter, we get only the light that we want. And so here we shine the light on the whole system because it's wide field. So the whole system is illuminated. And you see, if we have a laser, because the lasers are monochromatic, we do not need the excitation filter. Okay, so we have a mercury lamp or an LED, we have an excitation filter, but if we use a laser, no excitation filter, but it's the same thing. So we shine this light, which is a specific range of light that we have chosen, onto the sample, and the light goes through the dichroic mirror in here, and then it reaches the sample. So we have already seen the 4 roles of the dichroic mirror, so you've been very good at doing that. It sends the excitation light towards the sample, it sends the emission light towards the sensor, and it prevents the reflected light with typos from reaching the sensor, and also we can use it to split the light between different sensors. And here we go, our sample is completely illuminated. So we have the whole sample is illuminated, the whole field of view is illuminated at the same time. And this is why it's called wide field. Okay. So you can see in here an image that I took from this course, where you have the objective. So this is an upright microscope because the objective is above the stage. And in here, there is actually a solution of a fluorescent solution, a fluorescein. And when we shine the light on it, we can see the shape of the excitation light. And what you can see here is that the shape of the excitation light is like a big cylinder. Okay, so this is defocused. It's not like a spot. It's a defocused illumination. That's why it's called wide field. And the shape of the illumination is a cylinder. If we put a sample in here, what happens is that maybe we're focused in this plane, right? And this is what we're imaging. But everything here and everything here will also fluoresce because it's also excited. So the whole column is illuminated. So if you look in Z, you can see that there's actually a big chunk of the fish that is illuminated. So now that we have our system here, and then we have wide field illumination, so we see that we have the excitation happens at the XY plane, but also in Z. So we have excitation out of the focus as well. So now our samples is excited, and then it starts fluorescing, and the red fluorophore, for example, is the light is coming back, the emitted light is coming back and reaches the dichroic mirror, which in this case reflects the emitted light. And then we have an emission filter in here. And then we can see that we have defocused excitation. So this is defocused excitation. We also have defocused emission as well. So we have a lot of light that comes from everywhere in the sample. And what we do is that we put the camera there where the point spread function is formed. We put the camera and we record all the point spread functions at the same time with millions of pixels, and we get an image with the same number of pixels. Okay. Now, what happens if you want to say imaging in wide field is very similar to flash photography. So I have all my friends are there in a group, and I take my my phone and it's set to automatic flash. We're a bit in the dark. So then we have the flash that starts, and the whole group of friends is illuminated at the same time. and then the camera collects all the light at the same time, but it collects the light from the friends that are close to me and the friends that are far away from me as well. So everything, the whole volume is illuminated and imaged at the same time. And if we look at this image, this is exactly the same thing as any digital image. So we have pixels, and each pixel has an X and Y coordinate and one intensity, one global intensity. And the light comes from all the different Z. So it's a volume. If it's not the case, which you will see later, this is what it would look like. So you see, this is in wide field, and this would be if you do not have the light coming from all of them, so that you can see the difference. So now let's go to a sample, a microscope that works with detectors, so no camera. It works with detectors, a single point confocal, it's the same sample. It's also an inverted microscope. Now the sample is in the dark because we don't have any light, and we have lasers. Point confocals always work with lasers, so it's quite simple. We have lasers, and then we have a scanner. And the scanner has a direct mirror inside. And we have currently one laser on, it's the green laser, which will excite red fluorophores. And then we have this spot of light, just a tiny spot of light. The laser is highly concentrated on one spot, and we only illuminate this one spot. So that's why we call it single point, because there's only one point. And if you look again with this little cuvette full of fluorescein here, if you shine some light and you look at what it looks like, in this case, the shape of the illumination is a double inverted cone, as you can see here. Yeah? So we have a focused illumination. So the illumination is focused here, but we also have illumination above and below the focal plane. So the fish is excited a little bit everywhere, but it's focused in here at the focal plane, but we also have illumination above and below. So the excitation is a small XY spot and a double cone in Z. And this is to be compared with what we had for wide field illumination, which was like a cylinder. So here we have what happens is that the little spot of light starts fluorescing. We have green light, so we get red emitted light. And then this light goes back into the objective, or part of it goes back into the objective and reaches the scanner again. And the scanner is going to send the light towards the emission filter or the different type of detectors that you have. Some have emission filters, some do not. We will talk about this next week. And we can have between 2 and 34 single point detectors. Each of them only detects the light from one pixel, okay? And we will see all this next week. So then slowly we build the image point by point, line by line, like we have seen. And then what happens is that we have in here the focal planes. So this is the place where our sample is focused. And in here we have focused emission. So we focus excitation on the objective focal plane and focused emission. And now what we do is that we put a big metal plate on the emission light path just before the detectors, or actually inside the detectors. And we block the light almost completely, except for a little hole in the middle of this big metal plate. And some of the light only will go through the hole and be sent to the detectors. This is the pinhole. So we have here the objective focal plane, so we're in focus for the objective, and we have also the focal plane of the pinhole. And this is the reason why a confocal is called a confocal, because we have two conjugated focal planes, the one for the objective and the one for the pinholes. So we have a confocal, we must have a pinhole. If we have no pinhole on the microscope, we have no confocal, because we must have the two focal planes, one for the objective, one for the pinhole, and this is why it's called a confocal. So we have two conjugated planes. Okay, so what happens here is this pinhole, so we throw away lots of lights, but the pinhole is there to reduce the autofocus light that we have seen comes from this here and here. So above and below the focal plane, we have some fluorescence because we do excite with the double inverted code. And we throw this away thanks to the pinhole. So we remove this light, and we will see how that works. the scanner records the XY information for this particular point. Then the light, after going through the pinhole and the emission filter, will go to the detector, which records one intensity. So the detector only records the intensity. So we have one intensity per spot, and there we go, we build our image with one more spot, with one XY and one intensity. And then we move on. And how do we move on? The scanner is going to move the spot of light to a little bit further away. And this is the pixel size. So we choose, when we set our microscope, our point confocal, we choose the size of the pixel. It is the size where we jump. We jump to the next spot to measure. And it's up to us to decide the pixel size. We will discuss the pixel size a lot. Why do we choose one pixel size versus another? So this little new spot will fluoresce and the emitted light will go back into the scanner, and there the scanner is going to record X2 and Y2, and the emitted light will be sent through the pinhole, through the emission filter to the sensor that will record the new intensity, and the new spot will be built in the image, okay? So when we keep on going like this, we move again and we get X3, Y3 and the third intensity, and we continue, and then we build the whole image in that way, all right? Point by point, line by line. So how does that work? We have the pinhole rejects the out of focus light. Why does it do that, and how does it do that? So I remind you that when a fluorophore is a light source, and when it's excited, it emits light in all directions, and only part of this light makes it into the objective, and that is limited by the aperture of the objective. So the light goes into the objective, and thanks to diffraction and interference at the final image plane, which is where we image, we have a point spread function. And the point spread function is the image that the microscope forms of this very tiny point that is our fluorophore. And this is where we put our sensor and we record. And what does the point spread function look like? It looks, if you look at the image of just a tiny spot, you will see this bright blob and then concentrical rings. And if you do a Z stack through this and you look at it from the side, you will see a rugby guy, right? Well, we have a rugby ball here and then some rings, complex rings around. So if we look at the line profile here, this is what we see. We have now a familiar curve. This is the profile of the point spread function, the intensity profile of the point spread function in XY. Well, what the pinhole does is that it blocks the rings. So we allow only the bright blob to go to the sensor. So we put this big metal plate with, it's an iris basically. And in Z, this is what it looks like. So in the other dimension, so it blocks all the rings around, and it only allows the rugby ball to go through. This diameter from the first dark ring to the basically the diameter of the first dark ring here is called 1 area unit. And people who have worked on a point confocal will recognize the name because there's always a button there. Push that, you get 1 area unit. What does that mean? It means that you set the pinhole to the size of the first dark ring. Okay, so now you know what 1 area unit means. And in Z, it means that you are allowing only the main lobe to reach the detector. And so if you have a fluorophore that is slightly out of focus and a little bit away, it's very unlikely that it's going to be close enough and exactly just above so that its main lobe goes through. And so you're going to block the light of everything that is out of focus thanks to this trick of using a pinhole. Okay, so this is the fluorophore that is in focus, and this is the fluorophore that is out of focus. The light is going to be blocked by the pinhole. So the out of focus light is greatly reduced in a confocal or any system with pinholes. And only the light from the in-focus fluorophores is sent to the detectors. Whereas a wide field system, all the light coming from everywhere goes to the... So here are the similarities between wide field and single point confocals. In a wide field, and in both of them, we can have an upright or an inverted microscope. In both of them, we can have transmitted light imaging and fluorescence imaging, and both of them can be fully automated. So it's not the fact that the wide field system is sort of simple and manual, and the single point confocal is all automated. It's not the case at all. So we can have Z-stack on both of them, and we have a high optical resolution in XY on both of them. So we will have a workshop about that to demonstrate this. We can use fixed or live samples on both types of systems, and in both cases, the volume is bleached. But in the case of a wide field system, we have a cylinder shape of illumination, whereas on the single point confocal, we have a double code. Okay, so it's slightly different for that. We can do super resolution on both types of system. It's different techniques, but both of them can do super resolution. And now let's look at the differences. So in a wide field system, we use a camera. So we have millions of pixels. In general, we have only one camera, but it has millions of pixels. Whereas on a single point confocal, there's always a few detectors, minimum 2 detectors. That would be a very, very simple point confocal with only 2 detectors. But all of them image the same pixel. And then on the wide field system, we have like a snapshot, like flash photography. We illuminate the whole field of view and we collect the light from the whole field of view at the same time. Whereas on the single point confocal, we scan the sample point by point and line by line. So the illumination is a cylinder in wide field, and it's a double inverted code in single point confocal. It's focused on the single point confocal, and in wide field, it's wide. So that's why it's called wide field. So for that reason, in general, a wide field system is very fast, and a single point confocal is usually slow. But we'll talk about speed as well. In general, we can only do sequential imaging on the wide field system, which means that we would image the colors one after the next, not together. Whereas on the single point confocal, because we have several detectors, we can combine different colors and image them together. For example, the green and the far red, and the blue and the green, as long as we do not have any bleed through. So that allows us to go faster because we can image several colors at the same time. On the wide field system, we have no pinhole, and that means we are very photon efficient. Every single photon that is emitted, every single photon that is emitted by the sample is going to reach the camera. On the single point confocal, we have a pinhole, and we throw away a lot of light. Okay, we throw away everything that comes from the out of focus light. We only keep what is in focus. So that means that in a wide field system, all the Z levels are recorded at the same time. And if you have a slightly thick sample, it doesn't have to be thick. Even if it's just 1 cell diameter, you will anyway get some blur from the outer focal plane, whereas on the single point confocal, you get optical sectioning. So only 1 Z level is recorded. Okay, and the rest is thrown away. On the wide field system, if your sample is a little bit, a little bit thick, even 1 cell layer, you will get a lower contrast just because you get fluorescence from different Z levels. Whereas in a single point confocal, you get a high contrast because we throw away the light that comes from everywhere else, and we get high contrast even in thick samples. That means that the resolution of the objective can be really fully used on a single point confocal, but not fully used on a wide field system. And that is if we have a slightly thick sample. Wide field system, we have a low flexibility, and I will explain later. And on the single point confocal, one of the advantages of a single point confocal is the high flexibility. We can change the pinhole size, and we can change the pixel size the way we want. And on the wide field system, we have super high sensitivity, so we can measure single fluorophores. And in single point confocals, in most cases, you have a lower sensitivity than on a wide field system. And we will talk about this, the difference between cameras and detector, next week. And on the wide field system, we have no possibility to do spectral imaging, whereas on the single point confocal, because we have different detectors, we can do spectral imaging. So that's also very useful. And in general, a wide field system is simpler. It's simpler to maintain as well, and it doesn't require lots of alignment and everything, and it's cheaper compared to a single point confocal. So to boil this down, a wide field system is very fast, and a single point confocal is very flexible. There are other ways to remove the autofocus light. Removing the autofocus light is called doing an optical sectioning, because it looks like you have a section in a thick sample, even though you do not need to physically cut the sample. So it's called optical sectioning. And beside using a pinhole to throw away the autofocus light, there are other ways to achieve optical sectioning. One of them is a multiphoton microscopy, but we're not talking about this in this course. If you have a question about multiphoton, you're welcome to ask me. And then if you, another way is to use a multi-point confocal. And for this, we also use pinholes. So this is a camera-based confocal. So we will talk about that afterwards. And we can also use a light sheet system, which is also camera-based. And in this case, only the plane of focus is illuminated. It's the same here, only the plane of focus is illuminated. So there are different ways to get optical sectioning. Good. So now we are in the next little part of the lecture. I'm also a little bit early, so I think we are going to be we're going to take a tiny break, and